Good morning, and thank you for joining UCI Mind today for a live discussion and Q&A on whether we can change our genes and our risk for brain diseases, such as Alzheimer's. I'm Chelsea Cox, Associate Director of Education at UCI Mind, which is a part of a national network of Alzheimer's disease research centers designated by the National Institutes of Health. Here at UCI Mind, we're a collaboration of over 100 researchers, faculty, staff, and trainees who are working together along with colleagues around the globe to solve Alzheimer's disease and related dementias. If this is your first time joining us today, this episode is part of a monthly Q&A series where live viewers have the opportunity to ask questions of our experts at UCI Mind. And joining me today on the topic of epigenetics and the brain is Dr. Marcelo Wood. So I'm going to bring Dr. Wood on the screen here. Hi, Dr. Wood.
Hello, how are you?
How are you? Thanks so much for joining me today.
Absolutely, thank you for having me.
Now, before we delve into some of our questions, I'd like to briefly introduce Dr. Wood to our viewers. Dr. Wood is a professor and chair of neurobiology and behavior at the UCI School of Biological Sciences. He received his Bachelor of Science in Chemical Engineering from the University of Colorado at Boulder. From there, he changed fields to study the epigenetic mechanisms of cancer biology at Princeton University, where he also received his PhD. He then switched fields again to study the role of epigenetic mechanisms underlying long-term memory at the University of Pennsylvania. Now, here in his lab at UCI, he focuses on the role of epigenetic mechanisms in synaptic plasticity, normal long-term memory processes associated with drugs of abuse, and age-related memory impairments. So I see we have a few live viewers right now. Thank you to those who are joining us live. If this is your first time joining us, I'll get us started by asking Dr. Wood some questions on the topic for today. And then as you have questions that come up, please go ahead and type them there into the comments box accompanying the video, and Dr. Wood will address as many questions as he has time for. Please do remember that whatever you post on Facebook will be linked to your name and visible to others. Okay, so let's go ahead and get started, Dr. Wood, and thank you again for joining me today to talk about your research. And I was hoping that we could just first talk about epigenetics. What exactly is that word? What is epigenetics?
Sure, yeah, that's a great place to start. Epigenetics is a difficult word to wrap your head around and to define, even for scientists. We sometimes struggle exactly how to define this. But I'll first tell you what epigenetics is, and then I'll give you some examples of epigenetics in play so that you can get an idea of the power of how epigenetic mechanisms can regulate genes and cell function and brain function. So epigenetics means above your genome. So epi is above, genetics is your genome, so above your DNA. And so if you think of a single cell and all the chromosomes in that one cell, and you take all that DNA, that's about 6 feet of DNA. And you have to put that into this tiny microscopic nucleus of a cell. To get it inside that cell is a level of compaction of about 10,000 fold. That's a staggering number to think of. So if you take a piece of paper and you fold it in half, that's 1 fold. Fold it again, 2 fold. You can probably fold a piece of paper 6, 7 times, depending on how strong your forearms are. And a cell is folding up DNA about 10,000 fold to get it inside of a cell. And so that's a massive level of compaction. And to do that, a cell uses proteins called histones. And so the DNA and those histone proteins come together and compact your DNA. And so then there are chemical modifications on the DNA and those proteins. And those chemical modifications you can think of as salt and pepper and different spices. So if you have a plate of food and you add all your spices, salt and pepper, that flavors your food. And similarly, these chemical modifications add flavor to your DNA. And just like spices change the flavor of your food and really make a dish, similarly, these chemical modifications absolutely regulate your genes, where they're expressed, what kinds of cells they're expressed in, and they regulate cell function. And so the collection of all those chemical modifications or all of those spices that you've added, let's say, that's called your epigenome. Okay, so it's above the level of the DNA that you inherit from your mother and your father. That is the blueprint that makes us us. And the epigenome that's above that regulates how those genes are then expressed and where. So for example, a liver cell and a kidney cell, they have the exact same DNA. They're identical in your body. But a liver cell is very different than a kidney cell. How is that? How does that happen? It's because the epigenome regulates liver-specific genes in the liver and kidney-specific genes in the kidney. Another cool example of this, and one of my favorite aspects of epigenetics, is that many of those chemical modifications are metabolites, meaning they are derived from what our diet and our metabolism. And so, for example, in honeybees, a queen bee and a worker bee are genetically identical. Their DNA is identical, but they are very different kinds of bees, right? And so when a queen bee is a larva and it's growing up, it grows up in a larva cup with a special kind of food source that allows certain epigenomic changes to occur so that genes are turned on that allow that larva to become a queen bee instead of a worker bee. So the diet completely controls the development and the ultimate phenotype in the honeybee. And so similarly in our brain and throughout our body during development, in our adult stage, and when we age, our epigenome is controlling how genes are regulated and what they do and where they do it.
That's fascinating. So if I'm understanding correctly, it sounds like our environments and things can actually change the way that our DNA functions. And so what has your research revealed in terms of epigenetic changes as we age and as we get older?
Yeah, so when I started my lab here at UCI, I had, as you mentioned, I had started working as a graduate student in cancer biology. And in cancer biology, the entire field had been moving towards understanding how epigenetic mechanisms regulate cell function. And a lot of those epigenetic mechanisms had never been studied in the context of neurobiology, and in particular, never been studied with respect to learning and memory. And so when I started my lab here at UCI, I was surrounded by terrific learning and memory people who study neurodegenerative diseases. And my lab started trying to understand how these epigenetic mechanisms regulate gene expression that is required for long-term memory formation. So when you encode information from short-term memory that you can hold for seconds, minutes, into long-term memory where you're holding onto that information for a day, days, months, a lifetime, the neurons that encode that information have to change their structure and function in a persistent way. And so we have found that there are enzymes that regulate those chemical modifications that allow for gene expression to be turned on and off. And so, for example, in the young adult brain, there is an enzyme called histone deacetylase 3. We'll call it HDAC3. And it is involved in taking chromatin, the epigenome, and making it more tightly wrapped together. And so the genes that are there can't be turned on. And so normally, when we experience things, it takes a lot of signaling and a lot of activation to get those genes turned on so that we can encode that information into long-term memory. And HDAC3 is what kind of controls that. It's a molecular brake pad. It stops your memory from being, from encoding things. And so we found that if we manipulate HDAC3 we can then dramatically change how the brain is encoding information. So imagine if you have a car and you take off the brake, you can drive a lot better, a lot faster, more distance. Similarly with your brain, if we remove HDAC3 temporarily, we can encode sub-threshold learning events. So information that would not normally be encoded into short-term memory or long-term memory is now encoded into long-term memory when we release that HDAC3 brake pad. We can transform long-term memory into persistent forms of memory that last beyond normal long-term memory. When normal long-term memory would fail, these types of manipulations can generate a really long-lasting form of memory.
Wow. So one of the processes that's implicated in Alzheimer's disease is, of course, short-term memory. One of the first things that we see, one of the first symptoms. So, how does all of this translate to, how does the role of epigenetics translate to brain diseases such as Alzheimer's?
So first, we'll start with just the normal aging brain.
Okay.
And so in the normal aging brain, there's a hypothesis that my lab and others work on that the epigenome is becoming more repressive. And so it's harder for neurons in the brain to turn on the genes necessary for memory formation. And you can look at that in several different ways. You can manipulate it. We've manipulated it in the aging brain. And again, in the aging brain, if you release that HDAC3 molecular brake pad, the aging brain is able to encode long-term memory information just as easily as the young brain. You can change how the neurons talk to each other, the synaptic plasticity, so the communication, how they talk to each other. You can enhance it by changing that brake pad. In Alzheimer's disease, there are kind of two big things going on with respect to epigenetics. First, you have a lot of genes that are involved in Alzheimer's disease, like APP, presenilin-1, ApoE, TREM2. These might be familiar to the audience. And the chemical modifications that make up the epigenome around those genes have been altered. And so some of those genes are turned on way too high, some are not coming on when they should. And so those spices that we were talking about, the epigenome has changed now how those genes that are critically involved in Alzheimer's are being regulated. So that's one thing that's observed. The other thing is that you see misregulation of the enzymes that change those chemical modifications. So your salt and pepper shaker, the shaker itself has changed. It's either become this huge shaker or it's not working. So for example, in the cortex and the hippocampus, so regions associated with memory and plasticity necessary for information encoding, in humans and from AD patients, there's an increase in those some of the HDACs. And the HDACs form quite a large family of enzymes. And so there are specific ones that have inappropriately high levels of expression. And so you can imagine that if they're too high, then there's more of that HDAC brake pad involved, and genes are not being turned on in the way they should, and people have trouble then encoding information. And so you have those two dynamic processes involved.
Okay, so something has gone awry in a way in the Alzheimer's brain. You mentioned where chemical modifications have been altered, firstly, and then secondly, where there's been a misregulation somewhere. Mm-hmm. And I like your analogy of the salt and pepper shaker and then the ingredients that are inside them, and that kind of helps distill it for us. Now we actually have a question here from one of our viewers, so I'd like to address that first. Sure. What is the best way to change synaptic plasticity? By learning new languages or more education? So Dr. Wood, could you first explain to the audience what is synaptic plasticity? And then we can address some of the ways that might help.
Sure. Yep. So synaptic plasticity is a cellular mechanism, so that is thought to underlie aspects of learning and memory. And so if you have one neuron that's talking to another neuron, they'll reach out and touch each other, and they'll send chemical information and electrical information to one another. And that's how those neurons communicate. And there are massive patterns of that going on in different parts of the brain to encode information. And so as that changes and fluctuates, that's the plasticity. And it's called synaptic because the point at which those neurons almost touch each other physically and where they're sending across that information, that's called the synapse. And so synaptic plasticity refers to how the communication changes between neurons and networks of neurons in the brain. And so how to change that, all right? How to improve that, how to make it more healthy, longer lasting, more effective. There are ways that people are trying to develop pharmacological approaches. And so the more we understand about these mechanisms, the better we can find out which enzymes, so which salt or pepper shaker is going wrong and how do we pharmacologically fix it. When is the best time to do that? When you're just learning something, or maybe when you're trying to retrieve something. So there are different phases of memory that we need to understand how to change the plasticity underlying those. And then there are more natural ways. So exercise. Exercise is still probably one of the best ways in which one can change synaptic plasticity and memory function. Some of that work was pioneered by Dr. Carl Kottman here at UCI. My lab collaborates with him. We have a grant looking at the effects of exercise on memory. And here's the really cool thing about epigenetics. Your epigenome is a signal transduction platform. So what does that mean? It means that your epigenome encodes information about what you eat, how you sleep, your stress level. And like I said, all those chemical modifications that spice, they're metabolites. So they are also regulated by your diet and what you eat, pesticides that affect metabolism, things like that. And so your epigenome can encode experience. And so that's how people think that there's adaptive learning within species and within a generation. And so that's a fascinating idea, how some of that happens. And so exercise can be captured by the epigenome. It changes your epigenome. And if done in the right patterns and the right intensity, and that's what people are trying to understand and figure out now, you can completely change how synaptic plasticity occurs and how that synaptic plasticity then allows you to learn better.
Wow. So you mentioned, the importance of exercise, and kind of related to this conversation about how our lifestyle can influence disease. The next question from our viewer is, how can we regulate the enzymes in our body? And is this something that supplementation or nutrition can help with?
Yeah, so great question, right? And so there are pharmacological approaches being developed. So my lab works with several different companies in Boston where we know exactly which, for example, HDAC molecular brake pads are involved. And the new approaches in medicinal chemistry allow for the development of inhibitors that can go in and inhibit the brake pad. And that allows for gene expression to be facilitated, synaptic plasticity to be facilitated, and memory to be facilitated. So you can regulate those enzymes in that way. And people are trying to do that. And that has been used in the cancer field for decades. So for example, one of the first cancer therapeutics from the epigenetic side is a drug called SAHA. Some of you might recognize it as vorinostat. And there's a clinical trial being run out of Germany right now trying to understand how varinostat can improve HDAC regulation and gene expression for patients with mild Alzheimer's disease. From UCI, there's a discovery from Frank LaFurla's lab with Kim Green looking at nicotinamide. So nicotinamide can affect the cellular metabolism and energetics within the cell. And because those things are tied in tightly with metabolism, it also affects those kind of the HDAC family of enzymes and thus memory. And then there are supplements. And so there are some food supplements that people are trying to understand. And the problem is most of those don't pass the blood-brain barrier. And that's really the limitation in whether supplements are any good at all in the brain. They might work in the body and they might help with things like cancer for sure. But whether they can penetrate the blood-brain barrier and get to the brain where they need to be, that's the real question.
Mm-hmm. Now, just going back to a few things that you mentioned, you talked a bit about nicotinamide and the discovery in Dr. Frank LaFurla and Kim Green's lab. And I did just want to mention that that has now been translated into a clinical trial in people with mild cognitive impairment and early Alzheimer's disease to see if that can influence tau phosphorylation in the brain. And so, if you're interested in learning more about that, you can visit our website at mind.uci.edu. And that's just one of many examples of the amazing things happening in translational science here at UCI Mind, where discoveries are being made in the laboratories, like the labs of Dr. Wood and others, that are then being taken to patients and people who are suffering from these diseases. So it's pretty cool work. And then I wanted to go quickly back to HDAC3, Dr. Wood, and we have a question here from one of our viewers. Can HDAC3 be manipulated to help with short-term memory?
Yeah, that's a very good question, and one that everyone's still trying to wrap their heads around, not only HDAC3, but other HDACs and other enzymes that are involved in regulating your epigenome. And here's, and this is maybe too much detail, I'm not sure, but we'll go here anyway. So when memory formation is occurring, short-term memory is dependent on changes to synaptic plasticity, in a way that does not involve gene expression per se. But when that information is transformed from short-term memory into long-term memory, then you require gene expression. And so in our studies and other studies, we usually do not see an effect on short-term memory, but we always see an effect on long-term memory. And here's the strange thing. It looks like information that would have been encoded in short-term memory kind of bypasses the short-term memory mechanisms and gets encoded into long-term memory. And that's a almost heretical statement to say, because people think that memory formation is a linear process, a serial process. So first information comes in, it then goes into short-term memory. From short-term memory mechanisms, it goes straight to long-term memory mechanisms. But these data from my lab, other labs around the country and around the world have found that maybe there are parallel mechanisms in play, and you can push into one. And so information that could be encoded into short-term memory is actually being driven into the long-term memory mechanisms. And so it's a great question, very, very good question. And people are still trying to figure out exactly how that works.
Thank you. We have another question here from a viewer, Dr. Wood. How will in vitro brainoid structures accelerate our understanding of the neurobiology of learning?
Yeah, that's a great question too. And so one of the limitations in studying epigenetics, so I remember when I was a graduate student and we first started studying epigenetics in cancer. That was actually done in cell culture. So you take cells from patients with tumors, you can grow them in a petri dish, And then there were almost no tools to study the spices, the chemical modifications. You need certain tools to study that. And then as those tools became more and more available, then people were able to then start studying them in different contexts. And even now there's a new way to study this even in humans. You can look at HDAC density and distribution in the brain. You don't need post-mortem tissue. So the organoids and like brainoid preparations, they're fantastic because they can allow you to study more complicated interactions between different cell types and networks that are functioning together, rather than these totally isolated, reduced preparations that they're great because they give you an idea of specific mechanism, but then can you apply that to the more complicated in vivo brain is a different question. And then going from that into a human is even another level of difficulty. And so those organoid preparations are terrific for drug discovery and understanding these mechanisms in a much more complex system.
So I think we have a minute for just one more question, Dr. Wood, and I just wanted to end on what you think are the most important next steps in understanding the role of epigenetics in the brain and in Alzheimer's disease.
Mm-hmm. I think with respect to Alzheimer's disease and other human disorders, almost every single enzyme that regulates the epigenome, when mutated in humans, gives rise to either a pretty serious developmental disorder, an intellectual disability disorder, including autism, and heavily involved in neurodegenerative disorders. And it all comes down to when, where, and what genes. And so we have so little information about that. And really it takes an immense amount of funding to be able to traverse different species, develop the tools that need to be developed, and understand how these things are happening in different cell types. And some of that is just within the past few years, now finally at our fingertips. And so as the tools become better, our understanding will become better, and then we can really start to develop more targeted approaches, individualized approaches, and then we'll be able to make huge leaps forward in being able to deal with some of the consequences of the pathology of Alzheimer's disease as it relates to cognition.
All right, well, thank you so much, Dr. Wood. It was very exciting to talk with you about your research, and the things that you're doing are amazing. And thank you for taking the time to answer all of our questions today.
Absolutely, thank you.
Yeah, and thanks to all of you who attended with us live during this Facebook Live Q&A episode. I mentioned our website earlier, but again, if you want to learn more about Dr. Wood's research and the research that's happening at UCI Mind, you can visit our website at mind.uci.edu. And you can also read about daily updates about Alzheimer's research, as well as COVID-19 updates and resources, particularly for older adults, on our blog at mind.uci.edu.blog. I'll put that up here on the screen. And then if you're interested in learning about participating in research studies at UCI, you can visit UCI's Consent to Contact Registry website at c2c.uci.edu. Researchers at UCI and everywhere are always looking for more volunteers to participate in important studies like some of the ones that we talked about today. And lastly, I hope you'll join us next month for our next live Q&A episode on November 6th. I will be speaking with Dr. Daniel Nation. He's an associate professor in the School of Psychological Sciences at UCI, and he's going to be answering questions about how heart health impacts the brain. So we hope to see you then. Take care, and thanks very much for joining us.